Let me just see how it takes time. So thank you so much, Eric, for the introduction, and thank you everybody for coming. I'm very, very honored to be here today and very thankful for the organizers to have invited and have put together this symposium. So as Eric said, I'm going to talk about accelerating lineage discovery to document neotropical fern diversity. And just thinking about the topic of this symposium, New Horizons in the Study of Neotropical Floral, we don't have a lot of results yet, so you're going to see I'm going to be describing mainly this project and just reflecting on what we have learned in these 2 years. So before that, as Eric said, I am a botanist, a research botanist at the Botanical Research Institute of Texas. I was born and raised in Colombia, so that's why you see the 2 hearts here in this map. These are where my 2 hearts are. I do have smaller hearts in other places of the world, but so today I want to share with you a little bit, as I say, about this project, Ferns of Colombia. This is a collaborative initiative between some researchers in the USA and, of course, researchers in Colombia to better understand the diversity of ferns in this country. This work is supported by a National Science Foundation grant, a 4-year grant, and we are in the middle, so 2 years. So as I say, I'm going to talk a little bit about the project. I'm going to be telling you some stories or preliminary results that we are very excited about. And last, I do want to talk a little bit about the outreach, the science outreach possibilities that we have encountered, and also the implications of this work for conservation. So before I start, I really want to introduce this is a team effort. So here is what I call the core group. Mainly you can think about this. These are some people that are really directly being paid by the project. So the other 2 principal investigators are my colleagues and also fern specialists, Michael Sandu and Weston Testo. But we also have students in Colombia that are helping us also with a lot of the field work and things, but Susana is doing her master's also as part of this project in Colombia, and we also have grad students and assistants here. But this team is bigger, and so we work closely with numerous partners in Colombia. Here you can see some of the botanists that have been to the field with us or have collaborated with us. Some of them are interested in ferns, some of them are leaders in the botanical communities and just want to really facilitate our work. So we wouldn't have been able to do anything of what we are doing without our partners in Colombia. So here is Colombia, the northwestern tip of South America. And Colombia is one of those countries that have puzzled researchers trying to understand the diversity and evolution of neotropical biotas for a long time. And for many reasons, Colombia haven't been explored that much. And I'm not going to touch on that reasons here, but at least now there's a lot of botany happening there. The country have a lot of botanists, a lot of students interested in botany. And also we are being able, as researchers working in other places of the world, to go there and explore that diversity with them. So focusing on ferns and thinking about ferns, with more than 1,450 species in Colombia, Colombia harbors fern diversity that is unparalleled elsewhere in the Americas. So this is the country with more fern species in all the American continent. However, fern diversity in Colombia remains poorly known, and there are likely hundreds of species that remain either unreported for the country or are still awaiting scientific description. So nearly all, half of the species in the country have been collected only a handful of times. Most species have never been studied in an evolutionary context, and less than 1% of the Colombian ferns have been assessed for the IUCN Red List. So within this framework, the Ferns of Colombia project aims to improve humanity's understanding of how many fern species exist in Colombia, where they occur, and how many of them are threatened with extinction. So we are doing a lot of field work, and we are also doing a lot of curation of herbaria of museum collections, but we are also training the next generation of fern botanists and explorers. And last but not least, one of the objectives is really spread this interest, this love for ferns and for diversity and nature that I think all of us need to spread so that the general public and the communities really embrace this biodiversity, celebrate it, and help preserve it. So within this main aim, we are having 4 objectives that I'm just going to detail, and I'm going to be telling you a little bit of the preliminary results we have. So the first objective is addressing major gaps in our knowledge of Colombian fern diversity. So in order to improve understanding of the Colombian fern diversity, we are conducting field expeditions to understudied species-rich areas across Colombia. So at the beginning, when we got this grant, we were really happy. We were going to do six big expeditions. And as botanists, we love to do field expeditions, but these expeditions really involve extensive planning and coordination. And again, without the colleagues in Colombia, we would have never been able, or we would have never been able to do these expeditions. So I do want to go to the Emerald Forest with Alejandra. So here is a little bit of what we have done. So the 2 big stars, we have done 2 big expeditions, as you can see here, the 2 big yellow stars. And we also have done some smaller expeditions, either by part of the team or students that are working in those regions. And in 2 weeks, we are going to the other, we are going to start this other big expedition here in the south. So far, we have done more than 70 days of field work. So you probably know, so field expeditions, when you do them, they require, you have to be there before and after. So 70 days is really a field work. And we have collected around 1,300 new collections. We normally try to do 5 duplicates because some of the specimens stay in Colombia, some of the specimens come here, and we are studying them in both countries. We have thousands of photos of live plants. So only with 3 expeditions, with these 2 big expeditions and the other 4 smaller expeditions, I think we have work for the next 3 or 4 years to really process this material and really get to know what we are collecting and also do the taxonomic publications. So, but we still have 3 expeditions to go. So some of the results, I just want to share with you some of the things. We are still processing this material, but at least you can see we have collected species that have not been collected in the last 30 to 150 years. So here you see 2 examples. Luisma biovascularis and Parablechnus tubuli. Luisma has a really nice story because 15 years ago, when both Michael, who is the PI on this grant, too, and I were doing our PhDs, we went to the same place where we found this Luisma, and we were looking, Michael was really looking for this plant, and we couldn't find it. So this plant, Luisma biovascularis, was only known from the type till last year when we did this collection. So this time we went to a graduate student from Michael, Sarah Morris, and we were just wandering around collecting everything that we found. And all of a sudden Sarah says like, hey, Michael, I got this. And she shows the plant to Michael, and then there was a yelling. We use radios to call ourselves, so we don't go very far apart from each other, but we always are looking at different plants. So the radio thing, it was a big party because we found it after 15 years. So that was very nice. So we also are finding new records from Colombia. Here is Cyathea arsurgens and Elaphoglossum ornatiformis. Cyathea arsurgens was reported first for Ecuador and Elaphoglossum ornatiformis from Panama. I brought this Cyathea just for Pedro. I don't know if he's in the audience, but this is in the tree family, in a tree family genus, but the stem is lianescent. So this is a is in the tree family, but it's just not a tree fern, but it's very, very interesting, have a very particular habit. And we also, of course, are finding undescribed species that we are describing here is a Pterogramma and an Elaphoglossum. So the second objective is generating unified taxonomic resources for the ferns of Colombia. So a principal limitation to progress in the study of Colombian ferns diversity is the lack of unified and up-to-date taxonomic resources. So we are using an existing Symbiota portal that this was a big effort also funded by the NSF, where 17 herbaria and counting put their digitized collections of ferns. So we are really, sometimes when you go to a herbarium and you're looking at collections, you think like, oh, it's fast if I just put this data on my own database, and because I won't have to deal later with things that don't work in this portal. We are really trying to use this portal. We are really trying to make it better, to work with the developers, because we think, so we made this big effort, and we want to keep it up to date and working with that. So we are using this web portal as kind of like our manager system for the collections, old and new. And we have created a project-specific database that is actually in our webpage, and it's actually just, well, it's a snapshot of the Colombian collections within Pteridoportal. And so we hope that this streamlines our workflows, at least for taxonomic work. And we are also working with the partner institutions in Colombia to upload those data, the data that they have in their herbarium, in Pteridoportal. So to make them also an institution that participate in Pteridoportal in an effort to have everything together. This hasn't been as easy because some institutions don't want to leave their databases, but we are working with some of the main herbaria in Colombia to try to do that, because we really think this will be a very powerful tool for us and for Colombians studying their diversity. So we are also producing genomic resources for the ferns of Colombia. So DNA sequence data are critical for resolving taxonomic problems and placing Colombian fern diversity in an evolutionary context. But these data are really lacking for most Colombian fern species. So as part of this project, when we go to a field, we are developing this bank of silica dry tissue. And our objective is to generate target capture sequence data for all species of Colombian ferns. So at least one accession per fern. And we are getting there. I don't have any phylogenetics trees to show you, but we have around 50% already of the species, so that's around 700. Some of them are from other countries, so if Ecuador and Colombia share one species and we have that accession already, we count it as one that we have. So this is tied also with another NSF project that some of you might be familiar with, which was called GoFlag. So all these target capture sequence data are going to be the same as the ones that that project generated. And I don't have the number, but this project generated thousands of target sequence data for thousands of species of ferns. So, and last but not least, we want to combine all of this data that we are generating in the herbarium, in the field, and in the genomic to accelerate the pace of taxonomy and lineage discovery. So if you are trained as a classical taxonomy and systematist as I was, the accelerate part kind of like gives you, it's a very uncomfortable word. And when we were writing this project, I really dislike it, because I know that in face of climate change and a habitat loss, we really need to accelerate the pace on how we are describing our diversity. But it's really also true that you cannot rush what can be rushed. So revisionary taxonomy is a very important process. It needs time. You need time to look at the specimens, to look at the collections in the field, to really understand the variation and make your hypothesis. And so sometimes when I hear this word, I kind of like think about people trying to rush things that cannot be rushed. But it's true that you also can use, what we are trying is streamline all these resources and data that we have so that in the part of taxonomy, we can have the time to do a really good taxonomy. So we are leveraging all these resources and data, the taxonomic resources, so that's why we have this dedicated project database, so that we can minimize, for example, when we go to the herbaria, the input and the writing of data, and we are trying to get as many sequences as possible to have a reciprocal illumination in our taxonomic work. And of course, to accelerate the taxonomic, the pace of lineage discovery, we need to train more people who do that. That's another way. I mean, if you cannot rush because one person cannot just go to a herbarium and look at like artificial intelligence, you know, like just so that things get faster, you can train other people and collaborate with other people so that to try to make things a little bit faster. And so, as I say, a major component of this project is to generate data and develop the capacity necessary to document and describe unknown fern diversity. And so, because we really think there are a lot of, a high number of undescribed fern species in Colombia, we developed this strategy that we're calling next generation lineage discovery. So, and this has been, or is being done mainly by the students in the project. So Lucia is starting in the fall, and she's going to be working on Elaphoglossum. So we are working in 3 focal groups. Elaphoglossum by Lucia, Pleopeltis, but our PhD student at the University of Vermont, Sarah Morris, and Westesto is the principal investigator of this grant that is dealing with the main analysis and the complicated analysis that we are proposing to do that. So a little bit about this approach is, so what we are calling is, so within the framework of doing a taxonomic monograph, a classical taxonomic monograph, we are also willing to describe descriptive diversity, meaning mostly polyploids and hybrids. So those 2 processes are very important in fern evolution, and we really do not much about what is happening to the species in the tropics. So we mainly know hybridization and polyploid complexes in the temperate regions, and but in the tropics, where we have so many species, mostly in the neotropics and here in this region in Colombia, we really don't know much. So what we are proposing is to do this approach. So within the taxonomic monographs that we will be doing in these 3 groups that I just mentioned, we want to include identified specimens in our comprehensive phylogeny. So basically, what is affine, or all these specimens that you are uncomfortable with all the time when you are doing monographs, there are always specimens that you don't know where to put. And my advisor, Robin Moran, we used to say that we used to have a we really should have a burner at the outside of the herbarium so that we can burn all those specimens that don't make sense. But instead of burning them, we just want a piece of them now, and maybe try to include them in this target sequencing data. And using this genomic data to infer this polyploidy and the resolved hybrids with all these programs that we have now, HypePhaser, Enquire, and Grandpa, for example. And as I say, if you have questions about that, ask Wes, because I'm not a specialist. We are collaborating, and he's in charge of this. And so basically, we want to identify, within this taxonomic framework, identify putative undescribed taxa based on the criteria evaluated in steps 1 and 2, and then compare these putative undescribed taxa to similar species, as you do always with monographs. Go back to the specimens and look at them and try to see if what you are finding in the phylogenies coincides or not with the data, with the specimens, and then formally describe this new taxa in taxonomic publications. And we think this provides a different layer. Descriptive species, we know it's going to be a little bit crazy because we are starting with a lot of diversity, but we think this next generation lineage discovery will really give us many insights into the diversity of how the diversity has played or evolved within in the neotropics, right? And here you can see these are really preliminary. I just brought it. Wes is probably going to have a heart attack if he knows I'm showing this. But this is a phylogeny of Elaphoglossum, and as you can see, we are finding this is polyploids and hybrids in different colors. So I almost had a heart attack when I saw this phylogeny because yes, we were expecting some polyploidy and hybridization happening, but this is going to complicate a taxonomy that is already very complicated. But this is also very exciting. So everything that you see in color, so we are expecting that, for example, in this genus, we are going to have around 20% of the species being polyploids or hybrids. So coming back to how to accelerate the pace of taxonomy and lineage discovery, I want to finish this talk talking a little bit about the outreach and conservation. And I put it here, and I didn't put it in a different slide, because I think if you want to accelerate the pace in which we are describing these new taxa in highly diverse countries or regions, you need to start working with the community and collaborating with other people that do different things, right? And so conservation, we have been collaborating we are the taxonomic specialists, so we are using this taxonomy to support these conservation assessments that other people are doing. And in this case, we are working with Ana Maria Benavides, who is the leader of conservation at the Medellin Botanical Garden, and also with the Humboldt Institute, with the biomodels group. They have developed this beautiful platform where you can enter data that have been done for IUCN evaluations, and then you can predict ranges. And if you are interested, here is the webpage. They do have a lot of different species that they are working on. And if you have a preferred taxon that you like, also, I think they will be willing to work with you. So with Ana Maria, we have been working, they mostly are working with epiphytes. So they just finished evaluating 290 species of epiphytes, from which 60 were ferns. So we picked those 60 ferns together, and they were basically the groups that the team, the Ferns of Colombia team, are specialists on, so that we could start with something that we knew how to describe or we knew how to identify. Because as you probably have experienced, JBeef and these platforms that are showing and that are giving us the collections and that are out there, they need some taxonomic work. So we did 60 ferns, and so here you can see this is not only the ferns, but we found this is for all the species, but the patterns in ferns are very similar. So what you can see is that 10% of the species from our evaluations are critical in danger, and 23% are in danger. And if you can see, they are mostly in the most diverse regions of Colombia, which are the Andes, right? So these assessments are very important because they constitute the basis for the management plans that conservation botanists in Colombia do and also present for to the government. And so we're working with them, and it has been very excited. We want to do all the ferns. Some students in Colombia are really interested in this, so we hope we can keep doing, and instead of 60 ferns, have the 1,400 species that we have. We also have, as part of this outreach component, we do have a curated iNaturalist project that, as you can see, has a lot of observers, and we've been giving workshops and trying to participate in the local efforts that people are doing in Colombia, just to kind of tell them the best practices for taking pictures of ferns, and also so that they know that what they are taking, the pictures that they are taking, matter to us. So we normally meet once a month or so and curate these photos as much as possible. And to end this talk, we also have been trying to see how we can reach the community. So a little bit of what Alejandra was saying, we need to get our word out there. And as there was an article a couple of years ago saying that scientists can change the world, but we have to stop only talking to us. And I think now this probably sounds a little bit like repetitive. We always say it, and but we need to do it. But this takes time. So again, like taxonomists, we need time to do our work. Systematists need time to do their work. And who is going to tell the communities or the other people outside of the amazing things that we are doing? So these are two different projects that we have been doing, trying to combine art and science, and just to try to tell the message of the importance of diversity and fern research or research in general to the public, to stakeholders. And so here are the two projects. The first one is an art exhibition that is actually now on view at the Botanical Research Institute of Texas, where I work. And so this was a collaboration that we did with an artist and a photographer. She's a photography professor at the University of North Texas. And she was really interested in highlighting the artifacts that we use as researchers in biodiversity exploration. And Dornith actually worked a little bit with seed banks, and she had one of her works exhibit here at the Smithsonian a couple of years ago. So we were really lucky to be collaborating with her. And so here you can see the 3 parts of her exhibition, and it was very nice. We did a panel discussion for this exhibition, and the public really came. I mean, we are not in DC or in New York, so we are a smaller kind of town. So we have almost 200 people that came to the opening, and we have this panel discussion. People were really interested, not only in the art, but also they started asking questions about what conservation, field botany. So it was really great. And the second art and science project that I wanna share with you is this that we have done. So this happened first at the first expedition. We were really, I was sending pictures, and you can see here, so we work the editing and the original music of these videos. I'm just gonna put it one for you. Was done by Elena Yepes, who is a creative planner. So she works for big marks, and she does a lot of like marketing and pianist. And if you can see here, there's a common last name. So he was my brother. So I was sending these pictures to them whenever we had Wi-Fi, and they just came with the project, and they created these beautiful videos that I think are really great and are talking to different people about what we do. So here you go. So yeah, just to show people in a fun way and very artistic way some of the things that we do. And for those of you that don't speak Spanish, pensando and prensando is thinking and pressing. So they just were really playing with these words. And so again, trying to get our word out there. I always say that whoever invites me, either in Colombia or here in the States, to talk about ferns and our project, I'm always happy to do it. Also, the team has been doing a lot of different things. We have been giving workshops in Colombia and here in the US as well to students interested on ferns. We do have our own store with the merchandise of the Ferns of Colombia that if you wanna get any, and is there. It's for nonprofit. It's just for fun and for just getting the message out there. And to finish, and thinking about these new horizons in the study of the neotropical floras, I really like what Alejandra said, that we need to still do all botany with this modern framework, but we also need diversity. Doesn't only come from plants. We need diversity of approaches. We need different people working towards the same objectives. And so this is what I think New Horizons might mean for the study of neotropical floras. With that, I just want to thank all the people and, of course, the National Science Foundation for giving us the opportunity to really be able to focus on this, and the communities and all of our friends that have made this possible. With that, I think I don't have time for questions, right? Thank you. We can take 5 minutes of the break.
One quick question. So I am just amazed and fascinated with polyploidy and hybridization in ferns. It just blew my mind. But how much endemic species do you really have? And how much hybrid species are you able to so how much of the speciation is hybrid speciation? Or do you don't know?
Yeah, so about endemics, so ferns have less endemic species compared with flowering plants, and so it's less than 10%. We have, in the list, we have 110 species that are endemic species of ferns in Colombia. And those actually are the ones that we really want to evaluate for the IUCN criteria, right? To just really do the red list, because when you evaluate an endemic species, that's the evaluation, the word evaluation, right? And about hybrid and polyploidy in tropical ferns, we really don't know. We really don't know what is happening. There are some flora, some really good studies in other places, like in Japan, where we know that 20% of the species that we count as part of the floras are either of hybrid origin or polyploid origin. So we are expecting that at least 20% of the diversity that we have of ferns in Colombia is probably of a hybrid origin and polyploid origin. But also there is for sure a lot of cryptic diversity, and that's why we are using these 3 groups to really know what is happening. So more soon, hopefully in a couple of years, we can have percentages, at least for these little clades that we are evaluating, and hopefully if this approach works, which we think is going to work, hopefully a lot of people, a lot of students will like to just jump in and start looking at the groups that they work with.
All right, now you do have another opportunity to stretch your legs. We will reconvene at 4:15, and again in about 25 minutes.